you have some immediate questions. Also, if everyone could be muted until we get through, and if you do have a question, you can ask in the chat, or if you have a question towards the end, we can ask that question live. So we'll have plenty of time.
Oh, he must have got kicked out. The other thing I feel like I do have to mention before we go is the way we are taking attendance weekly now, because that's something that the state wants, is that we're looking to see that you were engaged in some format. So if you're in Zoom, like I wrote down everybody's name.
Hi, can everybody mute? Great. I'll mute all, so we'll just do that. Okay. Hey, guys. My name is Tim Sear. I'm with the Millipore Sigma. I'm a senior technology specialist, and Suman wanted to set up this call to talk about the Quantum. So I have the Quantum and the Milliflex Oasis, and there's Suman right there in the box. And Suman, I don't know if can you unmute yourself?
Hey there.
Great. Okay.
Yeah. So yeah, I am the sales specialist that covers the Pacific Northwest. So yeah, if you have any questions in the future, I'm the point person to contact, and I'm excited to have you all see a live demo of the Quantum. So long time coming.
Yeah, so everybody, today we've got two parts to this. The Quantum itself is set up for our old Milliflex system, but I have the new Milliflex system because no one's going to want the old system. So it's going to be really, really close to what it exactly would be. But the Quantum box is just not 100% set up. But the idea is basically all the same. So am I sharing this presentation?
Yes, I can
Great.
see it.
Okay, so the Quantum has been around for about 10 years. It's based off fluorescence, and it's based off of the Milliflex system, which is membrane filtration and fluorescence. The nice part is you still have the Milliflex, the Oasis, and you can still do membrane filtration with R2A, TSA, SDA, and these cool color-coded media. But the Quantum is just an addition. So when you do this, you don't have to worry about validating a whole different membrane filtration method. You can just use the same exact method and add this this fluorescence piece on top. It's totally non-destructive, and it also can be used with any ID option, even the latest Molotov. So it's simple in that all organisms can accept this stain, and the stain goes in, but it doesn't come out. So what happens is the stain is absorbed by the organism, yeast, mold, or bacteria. And as the stain is ingested through a specific pore, the cell with an esterase will cleave that stain. And that's what will make that stain unable to be ejected by the cell. And that's what will make the stain fluoresce. And so it's only visible in the actual cell and not outside of the cell, because the esterase needs to cleave that stain. And so obviously the more colonies you have, the bigger spot you'll see. And the spot itself could be very, very small. And that's really the power of the Quantum, is you really want to be able to see spots with fluorescence before you would see them with your eye. Again, this is the old Milliflex. So you would do a normal membrane filtration, a standard incubation temperature. But what we're doing is reducing the time. And that's the validation of it. You're going to have to figure out the time frame. And that's the bulk of the work with the Quantum, is figuring out the time frame. So it's just based off of LED lighting up a fluorescent marker. That's all it is. There's nothing technically that someone would have to really study to understand how to use the system. It's very simple to use, and it's based off of fluorescence, which is not an old technology, but we combine the LED and the cleaving of this particular stain with the esterase to create a powerful detection system. So there's a slight picture of what it would look like with the camera. And then there's 10 colonies there, and then the software would count those as well. Another photo example. And I'm going to kind of run through this presentation. This is going to be a live demo, but I just wanted to show a little bit about it before we started getting into it. The removal rack is another thing that you'll need, because when we use the filters and the media, they're going to be combined. And in order to stain this filter, you're going to have to remove it from the media, and you'll see how that's done in a minute. Full ID option. This was cool. I like this slide. Let me try to expand it a little bit. If you look, you'll see that all the colonies here grew over here. And so it goes to show with any rapid method, you want to have equivalence, they say, of 70%. In a quantification method, we really shoot for 95. And that means that in something like this, where you see a tiny little colony, that would show up, or maybe one that may show up here later. You'll never, ever, between two different plates, you'll never, ever get the exact numbers that you're looking. It's microbiology. You'll never get 100% agreement because the samples are different. So in this case, we stained and then we grew them out and showed that they were identical. But in microbiology, with your validation studies, that's why we do statistics, just to show that they're equivalent in microbiology. Again, equivalent with all ID systems, or excuse me, compatible with all ID systems. And we did, like I said, we did MALDI-TOF. So this is really just a visible. This filtration, incubating, and then you count at 5 to 7 days. Here, we're just reducing the incubation step, and we're counting at 1 day or 2 days instead of 5 to 7 days. Even upwards of 3 days could save people a lot of time if they're really looking for some odd bugs. The hardware's pretty basic, and you'll see it, so I don't need to go over it because I'd like you to see it. And anything that's filterable and you don't have interference can work on the Quantum. So if you're doing in-process, even investigations, the Quantum is a really, really good tool. So we tested these bugs, and we tested some sterile water with some ATCC. Now, ATCC are simple. Everybody should be able to get those, and we did. But we also did some environmental strains, which we were able to get at 30 hours on R2A. And we did some more slow-growing strains that we were able to get within 30 hours. And a lot of these are environmental bugs, which just goes to show, when you can do that, that's what you want to show in a validation. You want to show that you can get the tough bugs, because anybody can do E. coli and Pseudomonas. Those are the easy ones. And I like this slide too. We got a mammalian cell culture, and we showed that we were able to get Bacillus at 8 hours, all the way up to P. acnes at 48 hours. So it just shows the power of the system. And most people don't do same day. We've had some people try, and they've had varying success, 8-hour testing, 10-hour testing. If you're specifically looking for Bacillus or something like a staph, it could work. If you're looking kind of for general population, people are going anywhere between 24 and 30 hours. And then if you really are looking, you want to find P. acnes, 48 hours is a good time frame. And we got full services. We can do a feasibility study for you. We can do come on site and help you set it up and do a feasibility study, do a demo in your lab. We could use live bugs, any of that. We have full IQ, OQ, even PQ support, full training. We don't do where you would buy the system, then you have to buy the training, and then you have to buy the validation, and then you have to buy the retraining. We don't do that. I'm here in California, and if you guys were to buy a system and get the validation, then we would always be there to help you train. We're not interested. We want you to use the product, not constantly be trained on the product. So that's that. And now I'm going to, it's the presentation part. Any questions on the presentation part of it right now? Because that's over.
Yeah, this is Roy. Could you
Hi.
walk through the kind of the incubation again? So when you talk about 30-minute and then you talk about 30 hours, I guess is that 2 different incubation steps?
Yeah, so there are, and that's what I was going to get into with the demo part, but no problem. That can be a little confusing. The 30 minutes is for the stain to get taken in by the cell and cleaved. So once you're done with your membrane filtration and you incubate this for 30 hours, then you're going to stain it and incubate this. After after we took the media off, you're going to incubate the next step for 30 minutes.
Gotcha.
Great.
Hi, Timothy.
Yes.
This is Elia Sanchez from
Oh, hi,
Allergy.
Elia.
Hi, I have a question on the mammalian cell culture slide that you show us.
Yes.
What is making it specific to bacterial cells when you're showing the fluorescence, and were those cultures made of mammalian cell culture actually?
Yeah.
Or
Good question. We have a lysis buffer that we do use.
okay.
Yeah. that's
So you
specific
treat
to
your sample
mammalian.
first.
Yeah, we did treat the sample with a lysis buffer in that case. In that particular case, water, obviously you wouldn't need it. Product, most likely you wouldn't need it unless it is some sort of cellular based product where there is an esterase that could cause that could cause, basically it turns the whole thing into a flashlight because there's so much material on the filter.
Okay, got it. Thank you.
Great. Super.
Tim, what else is known to interfere?
You know, sometimes the titer of the cells are so high that it won't filter. That can cause problems because we just can't get it to filter at all. So filterability can be high, like maybe 10 to the 12, something like that. Other components of media can cause interference. Like we can't use McConkey agar because McConkey agar will interfere with the staining. But generally, if it's filterable, we haven't ran into too many cases where we had background interference, unless it was mammalian, and then we just use the lysis buffer.
Okay, thank you.
Yep. Super. I'm going to show the Oasis now. Great. Can you see that? Suman, can you see that?
Yes, it's
Great.
not right side up though. So
What's that?
the camera's not right side up. It's
Sideways.
sideways, yeah.
Oh. Oh. Let's see.
Tim,
I don't
if
see it yet.
you-
I just see the conclusion slide.
Oh, okay. Let me stop presenting then.
Oh yes, yeah.
Great. Ah, is that better?
Still sideways.
But still sideways. You need to
Sideways.
Like, yeah, rotate 90 degrees.
almost there, almost there.
I know this is me now.
There
Yeah.
you go.
Okay.
you
No
go.
problem. I'll just hold it. No problem.
Well, now I'm seeing Suman.
Oh, I think I have to mute.
What's that? Sorry, guys. There. Is that better?
Yes, that's better.
Okay, great. Sorry, this is how I usually do it, and I haven't had that problem before, but we'll improvise. Super, we good?
Yeah, I'm sorry. I'm not seeing anything. I see a black screen now. I don't know what other people are seeing.
Okay. Should be sharing. Can everybody else see the pump, this pump?
Yes.
I can see it. I can see it.
Okay.
I
Gosh,
can't see it.
Will, oh boy. You know what, maybe I'll do this. How about if I do this? Sorry guys, this normally worked in the past, but I'm gonna do this. I've done a few of these, so we haven't had a problem with it.
Right.
That's way better.
That's much better.
Backup plan. Got to have a backup plan. Okay, so this is the Milliflex Oasis, and these are the cassettes, and you'll see they're tied in. These come in a pack of 8, and they're different than our other Milliflex. And I know you guys have seen some of our Milliflex videos, our new Milliflex Oasis videos, but this Oasis is what was about 7 years in the making to deliver a product that you guys wanted. To start off, the lid is attached, and you can just close it when you filter. We've got the gradations. You can see when you fill, and I have some water here. You can see how the blue in the background shows up. Hopefully that kind of shows up and you can see what level you're at. So this funnel, when you filter, you just hit this little button here. And I'm filtering into So once I'm done, it's going to do a little dry out cycle. And I've got my media cassette here. This one particular is TSA. They're color coded. So this TSA is green. And at this time, I can take it off and put it onto my media. Now, this is probably the best part about the new Milliflex. You guys probably remember snapping funnels off, and that goes away. So again, I'm kind of demoing this close to the camera, so it's a little bit harder, but my lid just sort of, my lid is hinged. You can see the hinged lid. This just sort of pops off like that. So I pop that off as I'm on the bench, and all I do is just squeeze the funnel and it pops right off. So I don't have to do any cracking onto the media. I just squeeze, and that's a big difference. And then my lid goes on. So this is the differences between the two. Let's see, there we go. You can see this one's green here. They're the same size as far as filter. This lid is a little bit bigger. And again, you can see the color coded versus the other one that would be basically tan. So that's the Milliflex Oasis part of the filtration. And this little device just helps us remove this plastic piece that's on there. Now, let's see, we'll just do that here. Yeah, this little ring is kind of in the way. So here we have this ring that's also designed. It's green. You can get different color rings that are based off of the different products that you have. You might use, let me see, where's the gripper on this thing? Ah, there we go. There. That pulls it right off. So if let's say you had a pump that you wanted just for water, you could use a blue ring. If you wanted a product, you could use a green ring, and you can also label the pump themselves. And then finally on the Oasis here for sanitization, what you'll do is you'll put 50 mils of Sanitin in this syringe, and then you're going to inject the Sanitin into this funnel, and you can do 2 at the same time with this little device, and you're going to fill this funnel with 200 mils of water, and then as you press the sanitization, it's just going to go and sanitize both of these internal head. So technically, you don't need to do any other cleaning of this pump. There's no weight function like there was before. So this makes it so that you can do this daily. Some people do it before and after. It just depends on your product. It depends on your process. And so that just makes it a lot easier to sanitize. And you technically don't have to autoclave the head. People still will, I know, but you technically don't. It has a check valve there where you don't get any backflow. So, any questions on that part? Okay, let's go here to the quantum reagent. So I've got my plate, and I'm going to incubate that at 20 to 25, 30 to 35, whatever temperature works for your process. And then I'm going to pull it out of the incubator. And then I'm going to use this device. And I don't know if I'll be able to show it. Let's see if I can. Yeah, okay.
That works great.
Great. So what happens is I've got this set up, and I use this, and I slide it across, and it removes the media cassette from the filter. And this is why this matters, because look at, when I put my filter on the media, that filter is the filtration part, and the media is the media part. So I have to separate that and put it onto this liquid cassette. This liquid cassette is what holds my stain. So I separate my media here, all right, zip, and it removes it. And now I've got my liquid media cassette and my staining kit. Now, my staining kit consists of 3 bottles, and they're labeled 1, 2, and 3. So I take one, and I put it into 2, mix up 1 into 2, and put them both into 3. This bottle is for 48 tests. Okay? So at this time, I would, and I'm going to put this back over here, my pump, I have a different head. This is the staining head. So my other pump head is just for membrane filtration, and this one's for staining. So some people could have a pump just for staining, or you could have a pump here for membrane filtration and one for staining. It doesn't really matter. You could even have an old Milliflex pump if you wanted to for staining. It doesn't really matter. So I have my head and my liquid media cassette, and I put my liquid media cassette, and you'll see this has a Tyvek top, and I'm going to peel this Tyvek top off, so that's sterile. And again, as I have my, we're going to act like that's on there. I have a transfer pipette. It doesn't really matter what kind of pipette you use. You're going to put 1.7 mils. But what you don't want to do is you don't want to do too much because this will start to leak. Or you don't want to do too little because then you won't cover the entire area of the pad. Okay? So all I do, I get to about 2, and then I just put it on top of the pad. And if you can see that it's saturated the entire pad. Okay? Then, at that point, I've got my decapitated media. Remember my media cassette that doesn't have, or my filter that doesn't have the media on it. Okay, so then I'm going to put that on top. And then I hit start, and it's just going to draw that stain onto the filter. And this is an old one, so it ends up, that just ended up not really looking very good. But I have another one over here. So my final result is this cassette that has that liquid media cassette that's stained. That goes in the incubator for 30 minutes. Upside down. Usually you'll start to see colonies at about 5 minutes, but we validated 30 minutes. So let me move that out of the way. Now we'll operate over here. You guys can see that quantum. I'm going to turn it around. Okay, here's the quantum. It's a little box. So for, let's see if we can put a, here's a Milliflex cassette. So you can see it's not very big. It's not very big. So I pull this out of the incubator, and I'm going to put it onto my drawer. This drawer just slides in and out. And I'm going to slide it in. And as it lights up, can you guys see the colonies?
It's a little tough to see the colonies.
Okay, that's okay, because what we'll do, I have a connector, and that was, wasn't sure if that would work that good, so. Give me one moment here. Great. So you guys see that picture?
Yes, it looks lovely.
Super. So here on my software, I just hit capture. It's usually faster than that. And there's your picture.
Great.
So,
there's a question in the chat box.
Yeah.
What's the average total processing time from start to finish?
Well, hands-on time, a membrane filtration is, let me see if I still Am I still showing video, Suman?
Yes, I can still see you.
Okay, great. No, I just wanted to make sure I'm talking. So a membrane filtration, you can see that was really fast. So I pour my sample, filter, break down the media, put it in the incubator. One sample maybe take you 7 seconds, hands-on time, just for the filtration part. So once I'm done with all my filtrations, I move everything in the incubator. It incubates for, again, let's say 24 hours. So from that time, while it's incubating, I'm going to get my other pump head ready to remove my media and put my stain on. So I've got all my consumables for that. And from mixing my stain, that takes, you pull it out of the fridge, that takes less than 1 minute. And then as I remove my filter from the media and then get ready to put the stain on, you're talking, I pull it out, decapitate, put it on, another 15 seconds. So total hands-on time from start to finish is less than 1 minute per sample. You're still just waiting and still doing all the same things you would in a hood, doing all the same things you would in labeling, all of those things. That added staining step only takes, the vacuum where I pulled the vacuum on this to add the stain, that added step takes 3 seconds. You don't vacuum for very long. You just basically pull the filter onto the cassette. That's all that does. So I would say you're adding less than 30 seconds of hands-on time. There is that 30-minute incubation step, but you're going to get answers that day that you stain. So I hope that answered the question.
So in terms of shortening the length of time to get your result, the range of time would be, what would you say that range would be?
Like I said, some people have tried this at 8 hours because they're specifically looking for bacillus in a very investigatory phase. Other people who want to release water, they might do it at 48 hours. Some people want to look at product, have validated 22 hours. Anywhere in there. 72 is the max, because 72 you're really going to start to see a lot of colonies a lot of times if there's stuff there. Maybe not on water. So, if you feel like you have, I could show a picture here. Let me stop sharing for one moment. I'm going to show a picture. Okay. This was Pseudomonas in water at 18 hours. So you can see some of the colonies are pretty big, but some are pretty small, like this little guy at the bottom left. So for water, you could find, depending on your water system, you could find, if you're looking into whether you're releasing the water or even doing investigations, some people have done it at 22 hours. Other people, they might have bugs that have taken a lot longer. They go all the way out to 7 days. And you might end up saying, oh, well, you know what, for us, 48 hours. Most people aren't doing 72 for water. Most people, if they're looking for P. acnes in maybe a fermentation application, they might wait 72 hours, even incubate the plate in anaerobic conditions to help.
Great, thank you. There's a couple more questions in the chat box.
Okay.
One question, how many tests/samples can one person start in a 4-hour period?
It's going to depend. I'm going to share the quantum spot counter again. Sure. It's going to depend on the Milliflex. With this new Milliflex system, it's faster. The Oasis is faster and easier. So if you're talking a 4-hour session in a hood, having everything that you need, with the filtration going for less than 10 seconds, I mean, you could probably do 150 in 4-hour sessions. 40 an hour. I don't think that's especially with the dual head. And you can daisy chain these two, so you could make a 6-place Oasis kind of manifold, even though they're individually contained and you don't need vacuum. You could probably do 150 in a 4-hour window. And then the staining step would take even less than that because the staining step takes less than the filtration time.
Great, thank you. Another question is, what is the minimum sample volume that can be used? So I'm guessing that would be for the actual filtration step.
Yeah, here's the best part about membrane filtration. You could use 1/10 of a mil. You couldn't put that directly on the filter, but you could put 50 mils of fluid A and put your 0.1 mil onto the fluid A. Certainly. So the best part about membrane filtration is what you would think of as a dilution. You're still filtering everything that's going through that filter. So it's everything that's part of your product is 100% going through the filter.
Great, thank you. Are there any other questions before we move forward?
Yeah, does anybody want to talk? Anybody want to ask?
All right, well, guess there are no other questions as of now. Did you want to show how the software works a little bit, Tim?
Yeah, no problem.
Great.
Let me share here. Okay, so you'll always be able to look in the box, and I know this didn't work very well last time. So the camera's the camera. You'll always be able to look in the box to see the colonies. So even if you didn't use the software, you'd still be able to see the colonies and count. But if you did want the software, what it would do is when it reads, now you would use your mouse, and I'm going to click on every colony. And again, these are kind of dummy plates, so it's a little bit of a bad representation. You would usually see stuff like this, but without live bugs in my home. So there, I had 36 colonies and I counted them in about 15 seconds. Now you can save this picture and you can also print it out. What we won't do is we don't have the login, username. We particularly chose not to make this 21 CFR Part 11. We had a system that was fully 21 CFR Part 11 compliant, but people didn't like it for bioburden applications. They said, we don't need that amount of security for a bioburden application. So what we say is, this system will not save the count for you, but you can print the picture and you can put the information into your LIMS system and have that picture associated either digitally or printed out.
Great. That was actually the next question in the chat box is, can it transfer data to LIMS systems? And so you just answered that.
Yeah, no, and that was by design. And today's data integrity world is different than it was 10 years ago. I agree. But it just seemed like we got more pushback than we expected to have a system for bioburden and for water testing. People didn't want to validate, here's your IQ/OQ for the system, and here's your IQ/OQ for the software. And it just seemed like people didn't like that. So we ended up going the opposite direction.
Great.
Do you know what plants are doing then for interfaces with LIMS?
Yeah, they'll usually set it up to where if they save as, and they'll put it on some secure server that's a one-way. Or they'll print it.
So do you know if that's automating this?
No, it wouldn't be automated. Yeah, so there's automated detection systems that are out there, no doubt about it. There's automated viable sampling. There's a lot of automated that are out there, and the more the people that I talk to, they're just overwhelmed with the IQ/OQ of the software. And there's really no one good answer. So this system is a lot more flexible. It's not as high throughput as some of those other automated systems, but you're also going to have a lot less validation work.
Great, thank you, Tim. I have another question in the chat box. Are there simple to use thaw and spike microbe standards available to use for USP positive spike controls?
Yeah, I mean, there's lots of companies that have the E. coli or staph or bacillus, aspergillus, whichever bug you choose for a spike positive control. You know, you could put 100 CFU BioBall into 100 mils of fluid A, whatever their process is for rehydrating, and that would be just fine.
Great, thank you.
We haven't seen any organisms that don't show up, that don't use the esterase to cleave the stain. If I told you that we tested them all, probably everybody would leave the call right now, because of course we didn't test them all, but we haven't seen one that that doesn't stain, and we tested a lot.
Great. Are there any other questions?
Anybody want to see anything again? Any consumable? Any? This is a cool little feature. This doubles as a auger holder, with your final filter auger holder, and it snaps into place. So you could stack these on top of each other up to 5. So one little rack can hold 20 plates, and they're secure. You don't have to use tape or anything. And this can be done right in your hood, and you just take this rack right to the incubator. That was another feature that they built into this because of people using tape and insecure samples during bringing it to the incubator. That was one of the features that they built in.
Thanks for showing that, Tim. That's super nifty. I actually have another question in the chat box. Are the shortened incubation times compliant with USP?
Yeah, there are plenty of people who have validated this method with the FDA, and they are totally compliant with USP because you would use USP 1223. And so you would have to show equivalence, essentially. And that's what I mean about taking plate to plate. You would never say, I'm going to spike 10 to 100 CFU of Staph aureus in this fluid A and 10 to this 100 CFU Staph aureus in this, and expect to see 50 and 50. You would never expect that. That's why you do a bunch of samples over a time period, and you would choose. And it's not very hard, because if the product that you're using, let's say you're using some sort of aqueous eye drop, and you're using a 2 mil eye drop, and you put it into 50 mils of fluid A, and you spike it with environmental organisms. You spike it with staph. Staph and E. coli, those are what you would call your model validation. Does the system work as intended? Okay, I used all my 5 USP bugs. It works as intended, all within 24 hours, no problem. Then your working validation, how is the system actually going to work, is where you're going to test your environmental bugs that matter to you. And you'll say, great, I have a really odd Ralstonia out of my water system that takes 6 days to show up. Well, I'm going to take that Ralstonia and I'm going to run a set of dilution experiments, and I'm going to see at what level my lowest dilution can show up. So I might end up seeing my 10 CFU Ralstonia at 48 hours. And that might be your worst case scenario. And then you say, okay, well, everything else shows up for sure after 48 hours. So you'd say, okay, for my working validation, for my everyday day-to-day, I'm going to say that our reading window is between 48 and 53 hours or something like that. And that shows equivalence that when you run samples of that Ralstonia on R2A, again, we have R2A in this as well. When you run it on R2A without staining and you run it with staining, you'll end up at the same result. And even the USP talks about you can be 70% equivalent, and it's still approved. But we shoot for 95. We really do. We say, okay, if this plate has 100, we want at least, with over a lot of data, we want at least 95% equivalence between the two plates, because it's still growth based. And so you would then say, great, my validated method is now looking at R2A of my water system at 48 to 53 hours. And that's what you would submit to the FDA with the data. As long as you're showing equivalence, it's totally fine because you're still using TSA, SDA, and R2A as your medium.
Great, thanks, Tim. All right, do we have any other questions?
This the final result of this liquid is good for 7 days in the fridge. Also, you can aliquot it to freeze for 30 days. And this is a year shelf life as it stands, a year just in the fridge all by itself. But then also, if you freeze it, it'll be for 30 days. And then 7 days once you open it.
Great. And these plates, you can store them, you can keep those as well, right? Once they're stained.
I'm sorry, Sumo, what was that?
These plates, once they're stained and you've read them, you can keep those plates, right?
Yeah, once this plate, I'll grab this one that's, once they're stained, you can put them back in the incubator and continue to grow them. It's non-destructive, and so if you wanted to pick a colony to go to an ID system, you could go right off of here, or you could streak it out for isolation on TSA or some other medium.
Right. Thanks for clarifying.
And you can stain it up to 3 times, but most people only need to stain once. When you validate a method, there's really no reason to stain it more than once. You want to have a robust validation where you feel very comfortable with the method, that between that 24 and 28 hours or 48 and 53 hours, that you're going to get the result that you need.
Great. I have another question in the chat box. So this is from an individual who might be using this system more for research purposes. For the sample filtration step, is there any requirement to perform it in a clean lab? Will the environment cause contamination of the sample during the filtration step?
I mean, yeah, just on your application, for sure, people would do this in a hood. Yeah, they would do it in a hood. If you're just looking for a research use, you could use it anywhere. You know, anytime you're doing membrane filtration, you're opening this lid. And if you're opening this lid for a long time and you don't have a clean environment, you run the risk of contamination going into your sample. Now, from a microbiology perspective and just a research perspective, if I have this here in my dining room and I open it up, pour it in, close it, and it was open for 1.5 seconds, is that a high risk? Probably not. But if I'm looking at a specific count for my product and I need to release my product based on that, there's no way I would do something like that. So from a risk standpoint, it depends on what the application is. Also, if I did have particles come in, they're going to land on the filter. They're not going to proliferate before they get on the auger. So what will happen is that you'll see some sort of artifact contaminant. It will grow. There's no doubt it would grow. But so for an application wise, product and water testing, most, if not all, are doing it in a hood or maybe even an isolator for some products that people want to make sure that they have basically like a sterility test. And then other people, for research purposes, they might use it on a benchtop. But the more common is that it's done in a hood.
Great, thank you. All right, we're nearing the end. We have about 10 minutes left. Are there any further questions that we have for Tim while he's online?
Yeah, and if you'd like to talk, feel free to unmute and ask live.
I did have one other question about cost per test. You guys, do you have some sort of estimates of?
Yeah, it's roughly 14 to 15, depending on volume. The Milliflex Oasis itself is about 11 to 12. So the Quantum only adds a couple of dollars per test.
Okay. Thanks.
Yeah. The system itself, we are not one of those companies where, you say, what's the cost of your system? And then we have to contact you. No, this system up and validated with an on-site validation, full on-site validation, IQ, one pump, one reader, no extra equipment is about $45,000. So from a rapid method, that's pretty good deal considering some of the other rapid methods that are out there. Again, the flexibility you get as well. That's with full IQ on site, us doing the work for a week.
And then there's the, like a PQ would be then validating the incubation times, right?
That's really it. Yeah, you do your model PQ, which you got your 5 bugs, great. They'll grow, they'll work, no problem. And then basically your working PQ is really how it is, your operational PQ. And that's just your worst case bug. If you had a product and you knew that you had a specific staff that was a slower grower, sort of like a house resident organism, and you knew that was the worst case scenario, you would just validate the time frame that matches your standard result. And whenever you get to that time point, it could be 24 hours, it could be 48 hours, whatever it is. That's the experiment that you run, and you say, okay, I feel really confident that by 32 hours, I'm going to see everything I would see on my 5 to 7 day read.
Okay, any further questions? All right, I think we can wrap up then.
Great.
Awesome. Okay, as I said before, this was recorded, so I can send out the recording to everyone that attended this meeting, just so that you can have it to refer back to. But thank you everyone for joining. I hope you had all your questions answered and hope you got a better picture of the workflow. And if you have any other questions, just feel free to reach out to me.